been forever since I've used this one. But so now we're going to go on with the biosynthesis of the actual amino acids. Okay, so almost all plants and bacteria, they can synthesize all 20 amino acids. However, mammals can only make 11 of them. Well, it's 11 with a little asterisk of them. And so those 11 are the non-essential amino acids. The asterisk is tyrosine, and I'll explain why in just a moment. So we can only make 11 of them. All the others are called essential amino acids. We have to get those from our diet. Okay? Now, unless they specifically tell you otherwise, the book will only outline the bacterial form. Now, there's one later on that we won't get to today where they actually show you both bacteria versus the human, not human, well, it is human, but the mammalian form. This is to show you the context of the differences between them. Okay, but, and this is just a, well, I mean, I think that this is, let me see if I can remember how to. Oh yeah, it does. So this is just a figure to show how almost all of the amino acids, they come from some type of intermediate from the citric acid cycle, glycolysis, or the pentose phosphate pathway. Okay. Now, some of these are just within 1 or 2 steps. In fact, you already know how to make, for example, glutamate. I would expect you to know how to make glutamate and aspartate and pyruvate. But there are others that are really complicated. And so those are the ones that become essential for us because we just lack those enzymes in order for us to be able to make those amino acids. Turn this off. That's another thing that's annoying. All right, so what they do many times, how do I hide that? All right, so what they do many times is that they group it according to what the precursor was. So we can see here there are some that come directly from, or come from alpha-ketoglutarate, which is where we see glutamate and glutamine, which we already should know how to do those. And then we have some from, oxaloacetate, which was aspartate and asparagine. You should already know how to do those. Pyruvate makes alanine, and so on and so forth. Those that have the asterisk are those that are the essential amino acids that we have to get from our diet. Except you notice tyrosine's got the little cross. That's because as long as we have, if phenylalanine is an essential amino acid, as long as we have phenylalanine, we can make tyrosine. Okay, so that's why that's the shade of gray. And one common intermediate that you see, and I've got a slide on it coming up in just a moment, is PRPP, which stands for the 5-phosphoribosyl 1-pyrophosphate. Just easier to say PRPP. And it's just a common intermediate that you're going to see. I mean, you get it from the pentose phosphate pathway. Okay? And so it's also involved in the synthesis of nucleotides. And it should be pretty simple to see how it would be involved in the synthesis of nucleotides. It is a ribose. And like I said, it does come from the pentose phosphate pathway. Now, the way that it gets made is if you look down here, we have ribose 5-phosphate at the expense of an ATP. And this is one time where it's not the phosphate that gets transferred and it's not the AMP. So it's not being ampylated and it's not being phosphorylated, but it's being pyrophosphorylated. So it's an inorganic, well, what would become an inorganic pyrophosphate group that was added onto it. It's no longer inorganic since it's added onto an organic compound. But so if this was some type of just a phosphate that's being added, we would call it a kinase. But this one's not technically a traditional kinase in that sense. Instead, it's called a pyrophosphate kinase, which makes sense since it requires an ATP, but it's adding a pyrophosphate group and not a phosphate. So this is just ribose phosphate pyrophosphokinase. Hopefully the logic goes makes sense. Okay. Maybe. Kinase. Yes. All right. So that's just glutamate kinase. So now we have this, and the gamma just lets you know which of the carbons, because technically there are two carboxyl groups. Which of the carboxyl groups was phosphorylated? And just remember, it goes from the Greek lettering for alpha, beta, gamma, delta, and then epsilon would be the fifth one. So now we have NAD or NADH or NADPH that gets oxidized down to NADP or NAD. And so in doing that, we're going to make what's called glutamate gamma semialdehyde. And so, which is just a, it's an aldehyde. Okay, so since we've reduced this, what class or classes of enzymes have we seen that utilizes NADH before? What ones utilize NADH? Reductases can, and what's another class? I mean, it's the same thing, but sometimes they're called reductases, sometimes they're called, it's like the $5 word, dehydrogenases. And so if you're going to name this one, it would be called, and it's glutamate. Well, but if you said glutamyl phosphate dehydrogenase, I'd give it to you because people know exactly what you're talking about. Okay, but yes, it's a dehydrogenase. Now, the next step is actually non-enzymatic. And so if you think back to organic chemistry, remember you can have those, non-enzymatically, just like a sugar, can go in and attack itself. The scorpion can sting itself to make a cyclic compound. This is, same thing here, except it's just with the nitrogen being the nucleophile that can go in and attack that carbon. That's the acyl carbon. So that just happens on its own to make P5C, which is a mouthful for, it tells you where the double bond is, but that delta 1 purine 5 carboxylate, but you can call it P5C. And we're going to actually, I want to point out that you're going to see this P5C multiple times, okay? So we're going to see this again later on. So all this is a cyclic glutamate, in a way, semialdehyde. And then we have P5C at the expense of either NADH or NADPH being reduced to make proline. So what class or classes of enzymes could this one be? Pardon? Dehydrogenase again. And so if you're going to name this one, it would be P5C hydrogenase. And so once again, it's fine with me if you called it P5C pyrroline carboxylate dehydrogenase. And I would give you either the name of the enzyme or the name of the substrate. I wouldn't expect you to memorize the name pyrroline 5 carboxylate. I do expect you to know glutamate, since that's just, and proline, since those are just amino acids. Now we're going to actually go up here to the top, which unfortunately kind of got cut off. But let me see if I don't. So what does that do? Ooh, yeah, where's the undo? All right, so let's make it this color. So up here we have glutamate that's having an acetyl group added onto it at the expense of acetyl-CoA. So what kind of gets cut off up there is acetyl-CoA becomes coenzyme A. So we have CoAsh. And notice that it's being added on to the amino portion of the amino acid. So that's why it says it's N-acetylglutamine, or glutamate. So what class or classes of enzymes can this be? We've seen this kind of thing before. What's actually happening here? We're transferring it. Okay, and so many times it would be a transferase. This one's on synthase. Okay. But this is acetylglutamate synthase, which should make sense as whether you have two possibilities. One is a transferase and one is a synthase. And if you had told me the transferase, I'd give you both all the credit for it. But we made N-acetylglutamate. Now we're going to have the next enzyme. So I'm going to use my K. Now we have our next enzyme with the exclusive ATP that gets phosphorylated. And don't get scared by the gamma because it just lets you know which of the carboxyl groups, once again, was phosphorylated, since we have one on this end, which would be the number 1 end carbon, and then we have one on the gamma, so it's alpha, beta, delta, gamma. Alpha, beta, delta, gamma. Oh, just trust me, it's on that end. It's too early, it's Friday. So, what class or classes of enzymes can this be? It's a kinase. And so this is N-acetyl glutamate kinase. Okay, and for it, if I told you, since you know glutamate, and from organic chemistry you know what an acetyl group looks like, and the N is the locant, and there's only one nitrogen on glutamate, I would expect you to be able to put those pieces together. Now, the next step is synonymous to what we just saw on the other side, what we have just seen, I should say, on the other side with proline, in that utilizing an NADPH or an NADH, we're going to transform the carboxylic acid into an aldehyde. So what class or classes of enzymes could this one be? Dehydrogenase. And so if we were going to name it, we would name it N-acetyl glutamate dehydrogenase. Okay, we're not quite finished yet, but we're getting really close, in the sense that now what happens is we are just going to add in nitrogen, an amino group, onto that terminal aldehyde, okay, at the expense of a glutamate turning into an alpha-ketoglutarate. So we've seen this kind of reaction before, and for lack of time, it's just the aminotransferase. Technically, if you were going to name this one specifically, this would be N-acetylglutamate semialdehyde aminotransferase, because it's always named for the other, for whatever is not the glutamate, okay, the glutamate alpha-ketoglutarate. So that's why that's nothing new. Since it didn't all fit on one slide, I went ahead and moved it on. So we take this N-acetyl ornithine. This is where it should start to look really familiar, because the next step we're going to be making ornithine. And where have we seen ornithine before? The urea cycle. So this is the way that we're going to make arginine, is utilizing the urea cycle, but first we got to make it. And so first we have N-acetyl ornithine, and this one I won't even ask you to name it. It's got the special name. It's called N-acetyl ornithinase. So that's why that's probably one of the more difficult ones to remember. Or maybe it's not. It's to name initially. But we made ornithine. We're here within the urea cycle, which we've seen before. So ornithine, at the expense of a carbamoyl phosphate, becomes citrulline. And does anyone remember what this one was called? A mouthful one. Ornithine carbamoyl transferase. Then we have ATP and aspartate being utilized. So remember, in the urea cycle picture, if you think back in your mind's eye, remember it was the cycle part, and so they had multiple steps where they broke it down and they made it sort of the intermediate. But this one, we're making argininosuccinate, and so do you remember what that enzyme was? When you go from citrulline ultimately to argininosuccinate? Yes, this one would be a synthetase technically, because of the fact that it's going to have an ATP. And then finally, we have aminosuccinate, becomes arginine, part of the urea cycle. You kick off fumarate, where does fumarate go? Or where can it go? Citric acid cycle. And we ultimately have arginine. And what was this final enzyme called? And then we have argininosuccinase. So that's why, since it's not that difficult, the hardest part was because once you get to urea cycle, it's the same. And with the exception of the one that's a synthase within the acetyltransferase, they're relatively, they follow all the rules. See, that's why I don't know, it's a school the engine report. So that's why we'll have to figure this out. It's like what happens. But, yeah, so that's where I wanted to go ahead and